Hey guys, today we're going to talk about regulation of the cell cycle and how much DNA is in the cell at each point of the cell cycle. Cell division occurs during the mitotic phase, but this is only a short part of the cell's life cycle. For cells that are rapidly dividing, like our digestive tract or skin or cells in tissue culture, they divide about once every 24 hours, but cell division only takes about 1 hour out of that 24-hour period. The remaining time is called interphase. Interphase can be broken into 3 smaller segments. Interphase is split up into G1, or the first gap, S phase for DNA synthesis, and G2 for the second gap. We're going to start at G1 of the cell cycle. In this model, the organism is diploid. It has 2 of each chromosome, 2 long chromosomes and 2 short chromosomes. In diploid cells, homologous chromosomes are present. In a diploid cell in G1 of the cell cycle, there are 2 copies of every gene, 1 on each chromosome. So we can describe the total DNA content as 2N. The N refers to how many different kinds of chromosomes are present in the organism. In this model, there are 2 different kinds of chromosomes, a long chromosome and a short chromosome. So the value for N is 2. So the total number of chromosomes in this cell is 4. In humans, the value for N is 23. And so the total number of chromosomes in, say, one of your skin cells is 46. The next phase is S phase. The S is for synthesis of DNA. At the beginning of S phase, DNA replication begins, and the amount of DNA in the cell will double by the time S phase is over. At the end of S phase, this cell is still diploid. It still has 2 of each chromosome, or a homologous pair. But now every chromosome is made of 2 identical copies, the sister chromatids. There's twice as much DNA in the cell. There are now 4 copies of every gene. So the amount of DNA in the cell is 4N. Even though we've doubled the volume of DNA that's in the cell, there are still only 4 chromosomes present. The 2 identical copies of DNA produced by DNA replication, the sister chromatids, make up a single chromosome. Each chromosome is made of 2 chromatids, so there are still only 4 chromosomes present in the cell, 2 long ones and 2 short ones, but each chromosome is made of 2 chromatids. During G2 of the cell cycle, the cell continues to grow and it replicates the centrioles, which will aid in movement of the chromosomes during cell division. At the beginning of mitosis, the cells are still diploid and the DNA content is 4N. Mitosis will separate the sister chromatids into 2 different cells. Each cell that's produced at the end of mitosis is diploid and 2N once again, and it can enter G1 and continue to cycle through the cell cycle.